Now, the next topic is basically what all different types of numerical aperture objectives we use. Now, as I said, there are immersion objectives and there are non-immersion objectives. If we have air as a media, it is going to be a limiting factor. And if we want to increase the numerical aperture, along with the angular aperture increasing, we saw several factors there how to increase the angular aperture by decreasing the focal length, by using higher refractive index media, you are going to eventually increase your resolution. And the media that you are going to replace it with, it can be oil, water, or glycerol. But you cannot basically expect a single objective to work with all of these media, because they are, in terms of their physical and chemical properties, all of these mediums are extremely difficult. They're very diverse, and a single material or single lens basically cannot deal with all of it. However, the most preferable of this we saw is oil, because, and we'll come to that as to why. Consequently, the higher NA objectives are those which can actually have an immersion medium, and that's why all the objectives we are going to see in higher magnification that are used in today's modern day microscopy either use oil or air or glycerol as a suspension medium, as an immersion medium for them, in order to better resolve the subjects and get a higher NA objective. Basically, you are going to approach a higher numerical aperture value, and for that you need the immersion medium. So it's the need of an hour right now. And for using different immersion medium, actually, before going into what all immersion mediums are there and what not, we'll first see what all different type of objectives are there and how can you actually appreciate their presence. How can you recognize which objective to use depending upon your application. Now, a high magnification objective lens with low NA, that means, let's say the magnification of your lens is around let's say the magnification of the objective that I gave you is 100x. It has a numerical aperture value of around 0.95. In this case, it is of no good. Let's say I give you another objective which is of 100x and has a magnification 100x basically, and numerical aperture value is basically 1.45. Now in this case, it's which one is going to be used? You definitely should choose this objective, because we have seen multiple times in the previous slides that higher the numerical aperture, better will be the resolution. Now, if both of these configuration actually exist, which will be meaningful? Let's say you have another objective which is 63x and you have a numerical aperture of close to like 0.95. In that case, it would still be okay. But let's say if you have a 100x magnification, you would think that this objective is going to be better because it has same NA. So which one you will choose? 100x. However, that's not going to be the case, because you're going to magnify unanimously. However, you're not simultaneously going to increase your numerical aperture, which is the need. In order, we saw that merely increasing the magnification of your objective is not going to help you resolve your sample better. It's going to be basically both. You have to increase your magnification along with a significant increase in your numerical aperture to better visualize it. Am I saying that you will not be able to see anything with this? No, you will definitely be able to visualize with a higher magnification. However, how much that magnification will be of use to you without a significant increase in the numerical aperture is the question that you have to ask yourself. Okay. So we see that there are different kind of objectives. Now, many microscope companies offer objectives with high NA as possible. They want to go as high as they can possible to increase the numerical aperture so that they can provide this to us at a costlier price. And we are going to use this. Basically, we have to buy this if we want a better resolved image of our sample. So if you are in this position of buying a new lenses for your microscope, you should always consider buying those objectives which offer the greatest numerical aperture within your budget. Both of these things matters, both numerical aperture as well as the objective magnification along with your budget matters when you are actually going to go for a purchase of one of these. Okay, so now in order to examine the specimen or a biological specimen or any specimen that you want to at very high magnification using the microscope, there are a number of factors that you actually have to consider before you do that. The first and foremost factor is basically the resolution, which is controlled by the numerical aperture. Then you also have to see what is the working distance, which we abbreviated as WD. You have to see all of these factors, refractive index of the medium that is supported, basically your objective supports. As I told you, there are different refractive index, and there cannot be a single lens which can be used for all of these different medias. Therefore, there are separate objectives which can work at different immersion mediums. That, let's say there is a oil immersion medium, so the objective which we'll use for oil we cannot use for water and all those things. So we will have we have to make sure that we are using a compatible objective with a compatible immersion medium. And so we will briefly look at how you are going to use the immersion medium between the cover slip and objective front lens, which helps to increase your NA and basically eventually your resolution. Now, in addition, we will also consider the refractive index of air and the glass with which the slides and the cover slips are composed, and how an immersion medium is used to partially reduce the mismatch. When I'm going to come to a homogeneous immersion medium, I'm going to explain after that. After I'm done explaining this, I'm going to explain how and what a homogeneous medium can do to your resolution. We will also see some more practical tips towards oil immersion system, as well as the benefits of using water immersion objectives, especially when we are doing live cell imaging, and over that when we are using oil objectives, what are the advantages that you get when you are using oil as an immersion medium. But before all of that, you have to make informed decisions. Let's say I gave you a microscope to work on today, and there are several objectives, 4 to 5 objectives being placed there, and you want to actually visualize your sample with you chose to visualize your sample with a higher refractive index media like oil, and you want to know which objective you should use. For that, you should know what all markings and what all abbreviations are present on an objective to actually determine and actually basically identify which is the objective that you want to actually put your oil on and visualize your sample with. For that, let's see how objective markings are basically. Now, the first, you can see many markings being placed here, right? One by one, we'll see all of them. So the first and the very important marking, which is basically lateral magnification. It is showing 60x, so it's just basically a 60x objective. Then after that, it shows, you can see 1.40 as a number. Basically, that is the numerical aperture of your objective. Then just after that, there is a oil marking here, which is showing basically that the immersion medium that you're going to use is oil. Always before you actually start putting your oil, you should make sure whether your objective is actually amenable and compatible with using oil as a immersion medium. Now, your objective can be used for different purposes. It can be used for fluorescence microscopy, it can be used for phase contrast microscopy, it can be used for differential interference contrast microscopy and whatnot. So those kind of microscopy properties for which it can be specialized and can be used for are also mentioned. For example, here it is mentioned DIC. Now, there is a tube length which is mentioned here, and tube length is basically length of your entire objective, and inside when there are several lenses being there in the objective, where we will see this, and I'm trying to going to cover this topic much more in detail when I'm going to cover the anatomy of the objective itself. I'm going to cover what is tube length and how the different arrangements inside the lens is actually helpful to correct the aberrations that comes, because if you see there, here is a marking which is referred to as plan and epo. So basically it's showing that it is corrected for flat field and it's also corrected for aberrations. Now there are different type of aberrations which is possible, such as chromatic aberrations, there are spherical aberration and whatnot. So all of these to some extent can be corrected by adjusting certain extra lenses in your objective. Basically what I'm trying to say in brief is basically that your objective lens is not just a single lens. It's the front lens, that's the first one. Then there are other lenses in between which you put for different purposes. Some lenses can be to correct the aberration, some would be to actually magnify it more, some will be for flat correction, some will be for spherical corrections and whatnot. So all of this collectively makes your objective assembly of 5 to 6 lenses each of which have a purpose there. And in order to recognize that purpose, we want to know first why at the first place they are needed. And for that, I'm going to make another video. It's not a topic for this discussion. So we'll continue as to what all other markings are there. I think there is working distance here, which you can see. It's basically I think it's in millimeters. Then, or nanometers, I think. Then we have, it's showing that it's spring loaded refraction stopper. That means, let's say you have an objective here and you have reached this working distance of your specimen. If you go further, you can either damage the objective lens because it's going to hit your slide or a sample and a cover slip, and it can damage your, highly likely that it can damage your lens. So there is a mechanism which these manufacturers have developed, by which basically this has a spring-like capability to bend. Basically it can go inside if it's going to be applied with a physical pressure. This part basically can actually have a spring loaded there, and if you actually go and reach beyond the working distance, this part is going to, for a certain limits, is going to go inside, and it's going to it's type of a warning for you to actually go back, because you should never do that, because it's going to harm your objective big time and it's going to cost you a lot of money. Okay, so numerical aperture done, aberration correction done, immersion medium done. There are specialized optical properties done. Tube length, you can read, or I will also make a video to do that. Okay, there's another factor which is cover slip thickness. Now, the cover slip that you use for your mounting your sample onto your slide so that your objective can actually visualize it here, the thickness of the cover slip is very important, and for that thickness it is showing that what is the thickness which is amenable for your cover slip with respect to the working distance of your objective. We saw that the manufacturer has mentioned what type of correction, magnification can be done, aberration correction, and then here is a threading. Basically, threading is basically this kind of a concentric circle where you can actually fit it into the position in the tube lens, so that the next it can be formed as an assembly in association with your eyepiece, and that's how it's going to fit into your microscope. Okay, I hope all of this is very clear. Now let's see what all different type of objectives are available in this table. I have to try to put up this different type of objective magnification. Apart from that, what all different type of corrections are done. I will make a different video on these corrections, but for now you have to remember that the objective comes in 0.5, 1, 2, 4, 10, 20, 40, 40x oil, because this was air. Then you have 60x air, you have 60x oil, you have 100x oil, and then you have 150x air objective. But all of these basically are available commercially from different microscope companies, and you have to know that all of these do not make sense. They only make sense when they are in combination with certain chromatic aberration corrections, which is very important to visualize your sample with the objective that you are actually choosing. So you must read as to, when you're buying or you're actually using any objective also, you must know what all different kind of aberrations are there and how the different type of corrections are done in the form of flat field correction, in terms of basically achromat, whether it's apochromat, whether it's fluoride correction, and all those things. So all of these things eventually matters when you're actually coming down to your resolution. However, it's a separate topic of a discussion for another video, and I'm going to do that soon. So I hope you can basically appreciate that there are different magnifications available for the objective with different immersion medium. Here you can see that there is a 63x mixed immersion media objective. Then there is 63x oil immersion, 100x oil immersion, 63x water dipping objective. I'm going to eventually explain all of these objectives in detail to you, where I will be actually telling you how to use these objectives, how to clean these objectives when you are done using them, what all different factors need to be taken into account when different immersion mediums are used, which one is basically beneficial, and all those things. However, I would want you to appreciate the labelings here, which is one of the labeling is basically your correction color. The another labeling is basically which we missed here is a magnification color code. Now, if you see, the color code which is going to be in the form of this ring on your objective is very much standard. If you see, this is a Nikon objective. Now Nikon objective, and here we have a Zeiss objective. All the 63x objective basically have a code which is coded in terms of your blue color. Now there is another code here. When you have a immersion media, that also has a code. So if you're not, because when your objective will be mounted on your objective holder or objective rotator, it's basically a motorized rotator which has objective and you can change one after the other. When in case of an inverted microscope, the specimen is here, your slide is here, your stage is here, and your objective is somewhere here placed. Sometimes what happens, your objective is not even it's not completely visible to you, your markings and everything. So when you actually have to confirm, you can actually remember the code of the magnification as well as the code of the immersion medium. Both of these are very important and comes handy when you actually have to observe in an epi-illumination path where your light collection and the light illumination of your sample is done by the same lens. In that part, it comes very handy if you just notice what is the code of your basically the immersion media, what is the code of the magnification, basically what magnification it is, and all those things can be told to you in detail if you are going to keep these markings in mind and appreciate these markings, because this is basically a system that has been created throughout this period from when the microscopy started to develop and the manufacturer started to develop this microscope. So we should make a complete use of this and remember this, that don't use any type of immersion media with any other type of objective, because they are not always compatible with each other, as a result of which it can cause a huge damage to your objective, and it can cause you a lot of money. Okay, so after looking at what all other objectives are available that you can use for different immersion medium, we saw how objectives are basically named and what all markings exist on our objective, which gives us the indication as to how they should be used, what are the capabilities of the objectives, how and what kind of immersion media is to be used. Now let's come to immersion media. Now we know that refractive index is of great importance when it comes to actually filling the airspace between your objective and your sample, because this is the space where most of the rays is lost, which is coming from the specimen is lost because of very low refractive index of air, as a result of which more of the rays get diffracted and you cannot collect it. So this figure here exactly shows the same thing, where your objective lens is here, and if these rays which are present actually gets lost while the collection process, when the light source is illuminating your sample. Now this is, in order to avoid this, we thought of putting an immersion liquid. Now an immersion liquid is basically any liquid media which can be actually compatible to your objective, to your sample, and it should be placed and it should have one good factor, which is basically higher refractive index. Now what will happen when your light will emerge from the cover slip? It's going to see a media which has a refractive index higher than air, as a result of which the refraction of your beam of light is going to be less, as a result of which objective lens, the front focal length of the objective, can easily collect all the light, and therefore the light gathering capacity of your objective lens increases and eventually the numerical aperture increases, which is important to get high resolution images and detailed images of your specimen. Now there is a huge challenge in terms of when we are doing fluorescence microscopy, there is a huge challenge in terms of sample difference. Now let's imagine there are different type of samples that can be basically which are amenable for your fluorescence microscopy. Now when you are considering the refractive indices of the biological sample, you have to take into account, because you see there is a slide here, then above that is your sample in between, and above that is your cover slip. So here, when your light is actually illuminating your sample, the refraction or the deviation of the light from the path actually also depends on the refractive index of your sample. Now in fluorescence microscopy techniques, in this terms present us with a wide variety of challenges. For example, in conventional and confocal microscopy, we can identify three different or basically a broad class of specimen. It can be either animal, it can be plants or bacterial cells. Each of these has a wide variety of preparation method. They can be either prepared by mounting, they can be done live, they can be prepared by different kind of mounting medias, and all of these things are different. And we already can appreciate this thing who have studied basic biology in 10th, how animals, plants and bacterial cell components also at a fundamental molecular levels also differ a lot, which include, now all of these preparation methods will depend upon how your sample is, and if your sample is so diverse, your preparation method for these sample would also be diverse, which include whole mount, slice, culture, etc. Now, if you broadly subdivide these into three main types, the first type would be actually a living specimen. Now, these living specimen that you actually culture in your petri dish, which is basically, if you understand that if you want to do an animal cell culture, and if you are doing that, you actually culture those cells in the petri dish, right? And this petri dish that you have is basically the thickness of this is basically few micron. Now, if it is few micrometer thick, you think about it. The culture, which is the culture tissue, which is basically 30 micron, it's still transparent. There are several brain slices when someone wants to study, which is several hundred of micron thick with medium transparency. It is not very transparent. And when we talk about animals or plants, which are very thick specimen, when you take the tissue specimen of your plants or animals, it is very thick. Even it's often include a large variety of your pigments or non-transparent materials which actually block your light and is basically making your sample optically opaque. Now, any sample that has to be actually very well studied with fluorescence microscopy needs to be optically transparent, because light microscopy works on the principle of illuminating the sample and then collecting the light from the same specimen to actually gather the light and form the image out of it. Now, to maintain the viability, most of the living preparations that you are doing in case of your animal or animal cell culture, most living preparations are mounted in aqueous solution and buffers, because if you are actually keeping in these cells in the native aqueous or buffered condition, the integrity or the native integrity of these cells actually is maintained to the complete to actually a partial extent. However, this is the best way we have, because you need to maintain the temperature, that the physiological temperature that the cells will require. A physiological pH is of utmost importance for the cellular integrity. Apart from this, there are several other factors which are required in terms of nutrients and etc. Now, to maintain this viability, you basically can do one thing. You can actually keep the buffered, which is basically by doing the CO2, then the temperature maintenance. All of these things can be done. However, buffers in general have a refractive index which is close to water, somewhat like 1.33. Let's say suppose there were some cells in your petri and then you have kept it in a buffered saline, and this buffered saline basically has a pH of 1.33, which is very much close to water. Now, new methods of fixation such as cultured cells, tissue slices or thick specimen, as well as whole mount embryos that people are working on, the mounting media actually varies a lot. If you are doing a fixation based fixed actually samples, let's say if you want to fix the cell, there are different methods by which you can fix the cell and study the fixed cell or dead cells, or basically the preserved cell as we call it. You can do like methanol fixation, you can do formaldehyde fixation. One is cross-linking based method and the other is precipitation based method. Now, all of these methods differ from each other fundamentally. However, even after actually fix the sample, you need to keep the sample hydrated, and to do that, you have to actually mount your sample before imaging, and you have to do it with respect to some mounting media. Now, there are several commercially available mounting media available, such as movie wall, vector shield, or similar mixtures containing water and glycerol plus various other chemicals such as anti-fade and other preservative medias, like preservative substances, which will actually preserve your sample for a longer time. So if you actually save your sample by mounting it, you can save it for a longer time and can use it again and again after if you feel like you want to image the sample which you imaged last time 2 months back, you can still do that by preserving these samples by mounting them. Now, there are certain classically fixed specimens available, such as thick tissues and pieces of plants are actually embedded in resins. There is Canada balsam, or there is glycerin, gelatin, etc., which are usually used media for the plant material. Hence, the mounting media in itself, when you are using fixed sample, the mounting media itself has a large variety of, or basically a large range of all these substances that are named, have a large range of your thickness, as well as the sample is having another variability. However, I'm talking about the mounting media, which itself is very varied in terms of its refractive index, which ranges from one to other. Now, all of this matches with that of the glass for the mounting media. Now, as the immersion medium, selected oils are used, which again match the glass refractive index, that is 1.51. Now, why is this necessary? Why would why would the companies which are actually commercializing this mounting media are actually aiming to make the refractive index of this mounting media similar to glass, which is around 1.51 or near to that. Now, why would they aim that? What is there that they are achieving this? Why is not 1.4, 1.5 is not working, 1.3 is not okay? Why would they actually try to match that? Now, that's where basically the concept of homogeneous immersion media come. Now, the idea behind homogeneous immersion media is such that when you are actually using different refractive indices in your sample, in the sample preparation itself, then you have those many different kind of refraction episodes which the light will encounter while it is passing from one media to another. Now, although the different media, such as the material of the lens, the cover slip, as well as the glass, have different, are supposedly called to have a similar refractive index, but it is still similar, it is not identical, because the material out of which you slide your cover slip or your lens is made is fundamentally different. In addition to this, if you keep the specimen which is suspended in the mounting media, which itself has a different, altogether very huge difference in terms of its refractive index, it's going to actually cause a problem. Now, what would that problem be? Basically, the physical properties of the medium through which the light rays travel, whatever be it immersion liquid, be it cover glass, be it specimen mounting media, or be it glass side, all of these media have their physical property. Now, the physical properties of these medium basically decide the light rays actually to what degree the realized rays will be diffracted. We know the refractive index is numerical value without unit, which we know is determined to the extent to which light will refract when it is passing from one material to another. So all of these material being different, when it passes from one to another, it will refract. It is prone to refraction in that case. So when we know that air has a refractive index like that, and we actually used an immersion media which has a refractive index similar to that of the glasses that you are using there, glass material that you are using there, by doing that, you actually try to match the media of the glass there. Now, taking the difference into account, the purpose of immersion liquid is to match, or as closely as possible to resemble, the refractive index of the other media which are being in close contact with your sample, be it cover slip which is on top of the sample, or be it the basically the lens material and the refractive index of the lens, and also the bottom basically a slide or a dish on which your specimen is mounted. Therefore, increasing the amount of light rays, that is our purpose. Our purpose is to maximize the amount of light rays that can be taken or gathered by your objective in order to maximize your numerical aperture. One wish to do is to actually match or homogenize the entire refraction media that your light has to go through while it is actually illuminating your sample. Now, the refractive index of glass in which the specimen is mounted is actually 1.51. Now, subsequently, most of the immersion oil that are available today have a refractive index of 1.51 as well. Now, one of the problem in light microscopy actually is to overcome some of the limits of the optical resolution and to increase the numerical aperture of the system. That's what we are dealing with, right? It is very, very, very difficult to actually get prepared your high numerical aperture objectives. There are many corrections that has to be done to that objective, which actually increases the overall price and availability of these objective as a big trouble to us. Now, one of the Now we know that the increasing Na of the system is not that easy. You have to do several attempts. You have to actually attack this problem from different angles. You have to also try to increase the refractive index. You also want to increase the angular aperture. You also want to decrease the working distance. You also want to keep the objectives near as close as possible to the specimen. All of these things you have to do. Now we have already seen the concept of resolution and Na and how they are related. We will now examine immersion techniques available to microscopists which allow the image of specimen at high magnification without overcoming, actually also at the simultaneously they should also overcome the limits of resolution, right? They should not cause a problem. They should actually help us get the Na better so that we can get a better resolution at high magnification. Now this problem is very evident at high magnification because these high magnification objective have a very small working distance, and these are the magnification at which you really need very high numerical aperture so that actually you can study the detail of the system, because at this magnification only this detail can be visualized. However, if you don't have a numerical aperture that is actually able to visualize those detail, you will lack. So you want both the magnification as well as the resolution, which is going to come from numerical aperture and the detailing that comes along. Now, having an immersion liquid in place of air gap between the front of the lens and the objective, between the front lens of the objective and the cover glass of a specimen, which increases the resolution of the objective. Now, when light passes from one media to another, for example, let's say here it is passing from glass to air, it is reflected by the cover glass or actually blocked by the metal housing of your objective. You see here, there is a metal housing here. This is a metal case basically, and this is the lens of your objective. Now in this case, I would draw much more other rays also, then they will come here and they will divert here. So there will be a part of rays basically which is actually going out in the air because it's getting diffracted and getting lost, or there will be also the rays of light which are actually hitting the mounting or the metal mounting of your objective, as a result of which they get blocked, and this metal housing of your objective front lens do not contribute to the image formation. Now that's a problem, and that's what we are discussing for last 5 minutes. The purpose of immersion fluid is to decrease the amount of refraction and reflection of light from the specimen and increase the ability of your objective to capture this otherwise deviated light path, right? Your purpose is basically to put the immersion media so that all of these light can be actually put into the path of the lens, and they will be all eventually captured by your lens so that it can contribute to image formation. Now the ideal scenario is to create what is known as homogeneous immersion system. Now using this system, it is possible to actually achieve the maximum resolution and maximum numerical aperture, because you see here your immersion liquid has 1.5 Na, your cover glass is supposedly having 1.5, and specimen has a mounting media which has around 1.46, and it also ranges from 1.49. So that's almost a similar range, and then the glass slide also is made up of glass, so it has a refractive index of 1.51. So all of this somewhere or the other basically match your refractive index in a term that actually it is not much difference between these different media. So this is what we mean when we create a homogeneous refraction system. That means all the episode of refraction that is going to occur otherwise, you are trying to minimize all those refraction episodes, and thus you are actually matching the refraction or the refractive index of all the different media or all the different interfaces. You actually match the refraction of all those media and the refractive index and the Na of the front lens of the objective, so that most of the light can actually enter because of the presence of the immersion medium. Now here in principle all of these must have a similar refractive index. That's what we know, and this is what we call as homogeneous immersion system. Okay, so if we are actually clear with why we want to first of all introduce a immersion medium, and then when we want to immerse, actually want to put the immersion medium out there, we are going to have one more need, which is that all of these media that are going to actually offer the interface for refraction, all of these different media should offer more or less a similar refractive index, as a result of which it will create a homogeneous immersion system. Now after looking at all of this, let's go and see what all different type of immersion oils are available for light microscopy. Now, typically, I will be actually showing here some oils which are specifically offered by Nikon. Now, Nikon is basically synthesizing all of these oil, and most of these oils are actually developed and tested specifically for the use of Nikon objective. If you're going to go and search for the objective that your lab has also, you will be able to search for that. Because we are actually routinely using these type of refractive indices oils or the immersion liquids in our lab, I will actually briefly encounter and work on these. I have actually personal experience of working with these oils, and therefore I will be discussing all of these different type of oils which are basically provided by the Nikons. Now, before I can go into the detail of this, I would want to show you here that although all of them are oil immersion medium, that is, they are basically the oil which is offered, all of these oil also have different physical and chemical property, as a result of which they are suitable for some application and some of them are not suitable for other. We will discuss why some of them and what are the details which actually make them stand out than others, so that they can be used for a particular technique and others can be used for a different technique. Now, first of all, Nikon manufactures 4 different type of immersion oils, namely type N, type B, type F and silicone oil. Now, these oils have been developed and tested specifically by Nikon for the use of its own objective that it sells, and therefore their performance cannot be guaranteed for lenses manufactured by other companies. That's what is stated on Nikon's website. If you go here, you can read in more detail as to how they actually suggest it to be used and how they suggest it to be applied and everything else, which we also will see briefly. But when they are saying that this is specifically for the Nikon objectives, what I think they are trying to say is basically the refractive index and all the physiochemical properties are actually very much compatible with the lens material that Nikon is using in its high NA objective that I showed you. So in the oil immersion objective that I showed you, Nikon uses this kind of oil. I will show you what kind of oil, objectives, oil immersion objectives are actually supplied by Nikon, and they are of which microscopy technique they are applied of and other things in subsequent slides. But before that, if you use a non-Nikon oil, what will happen on the Nikon lenses? They say that the care must be taken to ensure that the refractive index and the dispersion value of the oil matches the property of the Nikon optics. So basically it's coming to the same point that your homogeneous medium, that the immersion medium should be homogeneous. It should actually match with the front lens of your objective and your oil also. So if these two actually have a good compatibility, that's the only thing that matter. As a result of which your light will enter and your imaging can be done very with a high resolution and details. Now, when we are saying that the dispersion value of the oil should match, it means that how much the oil allows basically the rays of light to disperse is also one of the property of light. Now, any mismatch here, you see, if any mismatch occur here, what will happen? Light will get a chance to actually have refraction episodes, which is basically going to lead to loss of light rays, either by actually moving into the media outside the medium, or it can actually lead to hit the mounting of the metal mounting of your casing, the metal casing of your objective, as a result of which eventually you will lose those rays and you will compromise on your performance. That's not what we want. Now, it also can lead to actually aberrations that the objective has, which is actually minimized by using the oils that the company provide with their objectives. Now, it is also important to note that oils should not be mixed with each other. All these different type of oils that I'm showing are actually having their own physiochemical properties, as a result of which you cannot mix them and cannot use a mixture of it. What happens if you use a mixture? Just think about it. If the physiochemical property of the type F oil and type B oil are extremely difficult, when you mix them, there is a mix-match of the refractive index, there is a mix-match of the temperature that they are stable at, there is a mix-match of the pH that they give, the mismatch between the physiochemical properties which actually help maintaining the drift correction. We will come to what drift correction is, but these important properties are taken into consideration when you are actually using the immersion media. because with great power with comes great responsibilities. You have to make sure that you are actually making a wise use of this immersion oil. Now let's go to one by one each oil, and we will see what application, basically to what they are actually applied for. Now talking about the type N oil, it's basically a general purpose oil. It's recommended for all different type of transmitted light microscopy. Now what do I mean by transmitted light microscopy? It's basically in which your objective is here, you have a sample placed here, and then your light, the condenser lens is here, and your light is coming from the light source, actually passing through your sample which is placed here, and the transmitted light actually, which is actually transmitted from the sample after it completely passes through it, is entering into the objective to form to go to the camera, and the image is formed. Now in this case, since the transmitted light from the objective from the object is actually able to be is actually used to visualize the image, it is called as transmitted light microscopy. And all the bright field, dark field, and DIC and phase contrast imaging, it is available to use because they are all the diffracted limited because all of them are transmitted light microscopy. Nikon suggests to use type N oil for this, which is a general purpose oil. Okay, now with this, they are also mentioning some squeeze bottles and all those things. I think you can go to their website, and if you're actually having these oils in your lab, you can actually read more about them, and before you actually try to use them. And we'll go to the next oil, which is type B oil. Now type B oil is basically compatible for the same purpose, the transmitted light microscopy. However, it is actually used for a long range experiments. Okay, now I have to bring in the concept of drift correction. So what happens? Suppose you have a epi elimination in your microscope. I have to draw this. Okay, so we'll take a break from what type of oil we had, and now we will discuss what is drift correction, because that is very important to understand what is the drift along z-axis and how oils can actually be used, which are compatible oils, to prevent that. Okay, so let's see. We have an objective here. Now this objective, as I told, is not a single lens. It has a first lens here, then it has certain other plano-convex lens, certain convex lens, biconcave lens. All of these things are there. I'm not going to go into the detail as to what lenses are there. I just named a few lenses which could be present there to correct various aberrations which are arising from single lens objective. So what happens if you have these many lenses in a block or metal casing which is made up of titanium? Entire assembly has a considerable weight. Now when you say they have a considerable weight, in case of epi elimination, your sample stage is above. And when your sample stage here, you would want to take this objective upside. Since let's say it is a 100x magnification objective and it has a numerical aperture of 1.49. Now this is a very good numerical aperture that an objective can have. These objectives are usually offered by Nikon, which are actually used for imaging in terms of single molecule imaging, or it can also be used for super resolution microscopy, which is done at TIRF, like total internal reflection microscopy, all those things. Okay, so in that case, what you would want to do is basically you have to take this microscope up towards the stage so that you can focus your sample. Now, since this is a high magnification sample, we have already discussed the high magnification sample objective has a working distance of around 0.13 to 0.16 micron. I hope I am correct here. No, it's actually mm. It's actually 130 to 160 micron. Okay, so such a less distance. In that case, if it is an oil immersion objective, which it usually is, basically these high NA objectives are oil immersion objectives. So when you will put the oil here, there will be a layer of oil which will be there on the objective. Now with this, it has to go and touch the sample so that it is in such a close proximity so that it can maintain this distance. Because why? This is a fixed distance that we were talking about when we were talking about the angular aperture and all those things. Basically, all these objectives work at a distance of 130 to 160 micrometer, because at only this distance they can perfectly focus your sample, and your sample is basically in correct Z plane to be visualized. So you need to have that distance or the working distance for them to be visualized. Now understand, this objective itself has a considerable weight. If it is lying at the bottom of the stage, basically it is in inverted form. In that case, it has on top of it a sample which is actually touching the cells. So let's say I actually take an eraser, I will rub the stage off. Let's suppose we actually focused our objective, and we are going to now draw our sample and how it will be in close contact, right? So let's say this is a situation and your objective is in close contact with your sample, and sample basically is what? The sample is, let's say, a fixed sample which is actually mounted by your with the cover slide, and there in between you have sample plus your mounting media. Now what will happen if you're actually imaging this system for a long time? Let's say you start to image this system for more than 20 to 30 minutes. Because of its heavy weight, or any which way, it will be acted upon by gravity. Now you think about it, the weight of glass slide, the weight of sample, plus basically the entire preparation, and this heavy titanium block which contains several lenses, it's going to be hugely affected. Now what will happen? If you're going to image for 20 to 30 minutes, let's say you have focused when you are starting the experiment, but as you go with your experiment for a longer period of time, like 20 to 30 minutes, what will happen? There will be a small drift. There will be a small change in the Z focus, or the distance at which your working distance is there. It is slowly going to come down because of the weight. Now, if that happens, your sample which you are visualizing will start to get blurry. Now this blurry sample is basically what you don't want, because you want the highest numerical aperture as well as high magnification can only actually give you the data which you want if it is completely properly focused. However, in the scenario, you won't be able to do that. As a result of which, what microscopic manufacturers have, companies have done is, they have developed something called as perfect focus system. Or PFS. Now, how this PFS was developed, what is the basically mechanism by which the microscope does that, is a topic for another video. But here I want to explain it with reference to why we are actually choosing the refraction media, basically the immersion media. So in this case, what happens if your PFS has to be activated? Basically, it's a function in a microscope. It's a function. Now, if I say it's a function in a microscope, what happens? If you activate this function, this entire assembly of the objective goes into autocorrelation mode, where it will know that you have fixed this. Basically, it will lock your Z axis. It will lock your Z, the position of objective at Z. What do I mean by Z? Z means along the axial direction. Now for any objective, the axial direction is basically moving up and down, and we know that the objective is a component in a microscope which has only axial movement. It cannot be actually moved in X and Y lateral direction. So this along the Z, that is upside or bottom, basically you can take it up and you can take it down, and that's the only movement which is available with the objective. And because it is prone to actually go down and go out of focus, it is given the system of perfect focus system. Now this perfect focus system actually works by knowing that you have actually locked some position. However, it actually tries to give some kind of a pressure from it tries to maintain and lock the position of your objective at a particular z angle, despite having basically a drift which it can have usually. So if you do not turn on this system and you try to image, what you will see, if you go live actually in your camera and you just choose one frame and perfectly focus it, after 30 minutes you actually try to come and see how is your focus, you will see there will be a small drift that you would be able to actually acknowledge that drift is there in your sample, and you would not have to like it. Actually, you would not like to have it, and still you have it, and if you don't enable the perfect focus system. So this is extremely important to use this perfect focus system when it comes to the microscopy which we are doing for a long time. Now, most of the microscopy, like super resolution microscopy, including single molecule localization microscopy techniques such as SPAM, STORM, etc., which are done at turf angle, needs a huge amount, like it basically takes a considerable time for actually acquiring a single image of STORM microscopy. Let's say if there are around 70,000 frames that you want to acquire, and out of those 70,000 frames, your exposure time is basically in milliseconds, let's say 20 milliseconds. We're actually thinking about 70,000 frames which has to be acquired one after the other at 20 millisecond exposure time. It's going to take at least 25 to 30 minutes to actually after focusing to actually acquire all the frames which will actually contribute to a single reconstruction of your super resolution microscopy imaging. In that, or in those scenarios, it is extremely necessary to use this system to actually avoid any drift in z direction. Otherwise, you won't be able to reconstruct the image and it will look like a drifted image along your z direction. Now, there are certain oil immersion media which are compatible with working with the perfect focus system, because the oil there helps these the optics as well as the camera optic, the objective optics to maintain that system and does not let this objective slip through it. Now, type B is where the type B is different from type N. In this case, it is compatible for variable length transmitted light microscopy, where you have to do the long hours of imaging with the transmitted light microscopy. However, when it comes to type F immersion liquid, it's basically designed to ensure highest image quality in case of fluorescence imaging, particularly the epifluorescence imaging, turf imaging, or any kind of a super resolution imaging can be done with this kind of a microscope. In our lab, actually, we do a STORM imaging using this oil itself, and it is basically also compatible with giving you a working with PFS, perfect focus system, and it also gives you maximum signal to noise ratio, because some oils are actually autofluorescent in nature. They will give you a background fluorescence which will lead to affect, which is basically going to any background fluorescence which is not a signal is going to affect your signal, as a result of which your background is going to increase and your signal to noise ratio is going to go down. Eventually, that's not the ideal thing we want in light microscopy, specifically in fluorescence microscopy, because we would want to avoid everything that is not a signal and take only and detect only few things that are the signal and make an image which has high signal to noise ratio, which actually contributed by the high numerical aperture objective. Now, it is actually compatible with PFS. It enables drift-free long time-lapse imaging and all those things that you would want to do with it when you actually try to apply it for long hours of imaging in case of live cell imaging, or in case of your super resolution microscopy. Now, that brings me to the last type of oil, which is a silicon immersion oil, which is basically used for deep observation of live specimen. Now, silicon oil has a refractive index of 1.40, which actually is matching with the cells, right? Because cells has a refractive index of about 1.38, and when you have actually used immersion media, basically, which is also having a not immersion media, basically when you have used mounting media which has the refractive index in this range, you would also want to use the immersion media which actually matches that, because we want to eventually homogenize all the interfaces that are prone to refraction, be it otherwise or towards this side of the light. But you want to homogenize that. In order to do that, you actually can use a silicon oil which exhibits virtually, basically in this case you will get high thermal compatibility, because even if you actually the sample which is for long hours of imaging is being used, even if it gets heated because of high laser intensity, the oil will not actually be it will not actually degrade or anything, as a result of which a sample integrity is maintained throughout the long hours of imaging and time-lapse experiment which you have to do for a very long time. Now it is also used basically these time-lapse experiment, you do not need to replenish the oil again and again, because that oil stays and it's not evaporating anytime soon. So that makes it extremely suitable for the observation of the specimen which are basically deeply basically which have deep or basically high along the, basically they are very broad along the z-axis or in axial direction. Now this was all about the type of the immersion oil which are available to us for use, supplied by Nikon. If you want to actually learn more about this, you can go to this link which I'm showing here and read more about these individual oils. If any of these you are going to use for application, I highly suggest you go there and read it, and before actually you start using it. It will make a great difference to your microscopy if you take small precautions beforehand, rather than just going blindly and using everything.